Hello everyone. This video is brought to you by ICCS Outreach Committee. We will briefly talk about tandem dyes in this video. In the earlier days, flow cytometry could only analyze limited parameters in a single run because the number of colors or the number of parameters were determined by the number of laser beams used. However, after the advent of tandem dyes, we are now able to measure several parameters in a run simultaneously. Tandem dyes were first introduced in the late 1980s. What are tandem dyes? Tandem dyes consist of two covalently bound fluorophores. One acts as a donor molecule and the other acts as an acceptor. When excited by the laser beam, the donor will transfer the absorbed energy to the acceptor, which will then emit the fluorescence. This process of transferring energy from the donor to acceptor is called fluorescence resonance energy transfer, also called the FRET. For example, we are all familiar with the tandem dye PE-Psi7. PE here is the donor, which transfers the energy it absorbed to Psi7, which then emits the fluorescence detected by the detectors. Tandem dyes therefore have the excitation properties of the donor and the emission properties of the acceptor. This allows us to use a single layer to simultaneously excite multiple fluorophores, which then emit at different wavelengths. For example, both PE and PE-Psi5 have the excitation property of PE, while PE will emit its fluorescence at 576 nanometer, which is shown here in yellow. PE-Psi5, which has the emission property of Psi5, will emit at 670 nanometers. This way, we can analyze two antibodies with the same laser beam in the same run. The commonly used tandem dyes fall under two major families, the phycobiliprotein family and brilliant violet family. The donors in phycobiliprotein family are usually PE, APC, and PerCP. This family requires an overlap of donor emission and the acceptor excitation wavelengths. They also have a protein component, which therefore makes them susceptible to donor emission and degradation. In the brilliant violet family, the donor is always BV421. This family does not require an overlap of donor emission and acceptor excitation spectrum. They also lack protein and therefore are less susceptible to denaturation. Here is an illustration of the brilliant violet family tandem dyes. First, let's learn about BV421. BV421 here shown as a blue polymer conjugated to an antibody gets excited by the violet laser and then it emits a blue light. The excitation and emission spectra are shown in this graph in purple and blue respectively. BV570 is a tandem dye which also has the donor BV421, which is again shown as the same blue repeating polymer, and the acceptor here is Psi3, which is shown in orange. BV570 is excited by the violet laser similar to BV421. This then transmits the energy to Psi3, which emits yellow-orange light. The excitation and emission spectrum here are shown in purple and orange respectively in this graph. So for both BV421 and 570, the excitation wavelength is the same, however the emission spectrum differs. There are several brilliant violet family tandem dyes that are all named after their emission wavelengths. For example, BV570 emits around 570 nanometers. There is BV605, which would emit around 605 nanometers. There is BV786, which would emit around 786 nanometers. Here is a list of the commonly used tandem dyes along with their excitation and emission spectrum. So you can see how the excitation max is the same for the dyes from each family, whereas their emission spectra differs. Of course, tandem dyes have limitations. Because tandem dyes are a combination of donor and acceptor, they can sometimes degrade or decouple, especially with prolonged exposure to light or higher temperature or even during fixation. And this can lead to dimmer expression or sometimes even spilling into other donor channels. The other thing we should remember is that when using a tandem dye, the ratio of the donor and acceptor fluorophore will vary with each batch. Hence, when we open a new batch, there could be differences in the compensation and the spectral overlap. So we should perform lot-to-lot comparisons religiously when using tandem dyes. Tandem dyes can also break down, causing false positive signals in the donor channel, and this might sometimes give an appearance of undercompensation or spillover. However, when we try to increase the compensation, that does not resolve the problem. So every time we use a tandem dye, we should keep this also in our mind and accordingly resolve it. Here is an example of the tandem dye breakdown causing false positive signals. In this example, we have APC bound to CD34 antibody, and we have APC H7, which is a tandem dye conjugated with CD19 antibody. APC H7 is a tandem dye with APC as the donor and H7 as the acceptor. So when this breaks down, we could see some spillover, or we could see some events in the APC channel. So it gives us an appearance there are some CD34 positive events, and also in the absence of a true CD34 positive population, this might look like as spillover or undercompensation. However, when we eliminate CD19 APC H7 and we just run CD34 APC, we don't see any events or signals in this channel. So this gives us a clue that this is due to tandem dye breakdown, and we should always remember this and also check our QCs for any hints of this happening. If you're more interested in learning about tandem dyes, here are some useful resources. Thank you for watching this.